6-day short-term course online on the broad theme of advanced materials for Aatmanirbhar Bharat. Welcome you, sir. Now stage is yours, sir.
Okay, so thank you very much, Dr. Bindu, and I'm thankful to the organizing committee and inviting me to share my work, we have been doing here. Actually, the work which I'm going to present was done in BHU because I recently joined here, so I had to start my research work. So the Bindu actually asked me, because my background is medicinal chemistry and chemical biologist, but she asked me, sir, can you give some lecture on material chemistry? I said yes, because we had worked on some molecule which has the material value, and those molecules were the carbohydrate based low molecular weight chelators. So I hope some of our audience or faculty who joined this meeting, they might be knowing what is the chelator and what is the value of low molecular weight chelators. And for the students, I should explain. So are you able to see my screen? Bindu, I think you are muted. So मेरी screen दिखाई पड़ रही सभी को?
Yes, yes.
जी sir, बिल्कुल दिखाई दे रही है sir. If you can make it full screen, I think that then it will be much better. Yeah, that's it, sir.
So the low molecular weight chelators, they are the compounds, they are small molecules which makes the gel either with the organic solvent. So if they makes the gel with organic solvent, they called organic chelators. And if they makes the gel with the water, then they call hydrogel. Typically, the low molecular weight chelators, they have molecular weight less than 1000. And what is the driving force, like how they makes the gel kind of property? So there are some non-covalent forces such as like hydrogen bonding or pi-pi stacking or van der Waals interactions. They makes a molecule to behave like say chelator. And the similar properties like hydrogen bonding, pi-pi stacking, and van der Waals interactions, they are the interactions also took place with the medicinal molecule. So because we were making a molecule and by targeting like medicinal value, but eventually we found that the molecule which we have developed, it having the gelation property. So if I represent the what is gel through the schematic diagram, if you see here, so these are the circle which is shown with the this blue color. This you can assume a organic molecule. Okay, and then these rectangles which are shown with the pink color, that you can just assume a solvent. This could be any let's say organic solvent, or this could be simply water. Okay, solvent molecule. So when these molecules come in contact with this organic molecule, they makes well-defined structures. So they got oriented in such a way. So the small molecules, they holds the solvent molecule, because solvent molecule, because this is liquids mostly, so either this is organic or water, this is liquid, so this freely flow. But when it makes the gel, so in like the picture shown here in the schematic picture, where it's got oriented in such a way that water molecules, they hold together along with the small molecule. So this property is called gelation, and this is the real picture where you see this is the hexane. Okay, so hexane is a solvent, is a liquid. So if you put that solvent in a like this kind of vial, and so this is free floating, but if it makes the gel, you see this is inverted vial, and still the hexane is not coming down because this got gelated. Okay, this particular property is called gelation. So Then the next slide. Yes. So what we have done, we designed a molecule which are carbohydrate based and which may have the function of gelation. As I told you earlier, I did actually these molecules was not originally designed for the gelation because I am the medicinal chemist, so we were making those molecules for the medicinal properties. But the molecule which have the medicinal property, the similar like molecular interactions have also happen with the gel, like van der Waals hydrogen bonding and pi stacking. That's why we got that particular material property of that molecule. So the carbohydrate based chelator, if I talk about, or sugar based chelator, they have many additional value. The first thing is they are biocompatible because carbohydrates. So carbohydrates is actually is our food. So they are not toxic. They are biocompatible. Second thing, they are relatively less expensive because the most abundant organic molecule in this planet. What it is? This is cellulose. So, and also plenty of glucose, plenty of starch is available on this planet. So, relatively, if you compare with other molecules, they are less expensive. The third important property the carbohydrate molecule have, they are biodegradable. Okay, so this is another additional property because gel kind of property. There are many molecules which may form the gel. But why we focus only the carbohydrate based gel? Because they have additional value like this. They are commercially available, they are eco-friendly because they are not toxic. And another very important property what carbohydrate molecule has, have their chiral. They have the chiral centers, and this the chirality actually helps these molecules to orient in a such a fashion in 3D space to acquire a predefined structures. Okay, so that helps into the self assembly of these molecules and the chirality of these molecules. So, and another property, they have the structural diversity because for the student, the glucose. So glucose has the four chiral centers, and the carbon 4 it has the equatorial hydroxy group, that is glucose. If you change this equatorial hydroxy to axial hydroxy, then that become galactose. And glucose is a sweet molecule because glucon D we used to take as energy booster. But when simple change into the stereochemistry, like from axial to equatorial, that molecule's property got totally changed. So galactose is not sweet at all. So that means in the carbohydrate molecule, we along with the help of this chiral diversity, we can also introduce to the structural diversity into the molecule. And as I mentioned, they are non-toxic. So these are some additional value of carbohydrates molecule is having. Therefore, we were interested to make the carbohydrate-based gelators. And there is a very nice actually review you can read on this Chemical Society review 5 years back has been published about the gelators. So this was the molecule which we have designed, and this is very simple molecule. This is a in carbohydrate term, you can say this is a monosaccharides because it has only one carbohydrate unit. And if you see this molecule, this NH acetate group here, which is called acetamide group, this beautifully has one hydrogen bonding donor, and as well as the acetyl group, the carbon it has acceptor. So donor acceptor in the same functional group. Then we have this free hydroxy, which is also participates into hydrogen bonding. Then we have this O-methyl group, which can potentially also make the hydrogen bonding. Then these are the two benzyl group, OBN, which can potentially make the pi-pi stacking with other molecules. So we thought about this molecule and which have hydrogen bonding sites as well as the pi stacking sites, and of course all the molecules they have weak van der Waals interactions anyway. So these two additional properties, these two important property of this molecule, we thought will be helpful to make the gel. So this is how we have synthesized this molecule. This is the synthesis, and the synthesis you can see uh, the glucose which is commercially available, and this is actually the glucosamine, not glucose, because we have NH acetate group at two positions. So this is glucosamine, which was glycosylated by using the amber light acidic reagent in methanol at 70 degrees centigrade. So the designing element for this synthesis is like when you do the glycosylation, this is called glycosylation reaction. So you simply we take methanol as a solvent as well as glycosyl. This would be uh, acceptor. Yes. So in acidic amber light reagent in the presence of methanol, when this molecule was stirred for 48 hours, what happens? We got a methyl group here. Instead of OH, this become OMe. And once the reaction is over, you simply filter it out, remove the reagent, and remaining methanol you can simply evaporate, and you will get almost 99% of methyl glycoside. Then the same material you can use for the next step by doing the selective tritylation, like and regioselective tritylation, the primary group by using pyridine. So you got O-trityl group here. So this is wrong mention. This would be OTR. So this is O-trityl at 6th position and O-methyl group at the position 1. So this was simple modification, like these two groups are selectively functionalized. Now you have these two free hydroxy group which was subjected for the benzylation reaction, and by using sodium hydride, and followed by, because we have to remove the trityl group which was removed by using 33% HBr, hydrobromic acid and acetic acid, and so this trityl group has gone. You have the benzyl group, and this compound 3 is got ready in 4 step. So within, like, actually this is 2 step, and actually this is a 4 step reaction for the tritylation, then for the glycosidation, then tritylation, followed by benzylation and the removal of ethyl group. And we can make this compound in gram quantity, compound number 3. Once we have this compound, we have started studying its gelation property. So we have used so many different organic solvent, like here you see from A, B, C, D, E, F, G, H, I, J. So these numbers are the different organic solvents. What they are, you can spell out. It's the bottom. So like A was the benzene, perfectly it's gelating the benzene. The toluene also all well gelated. Orthoxylene, metagylene, paragylene, mesitylene, and even importantly, if you see compound number this slide, while number G, the chlorinated solvent like 1,2-dichlorobenzene or chlorobenzene or even bromobenzene. So they were all well gelated. So I is the idobenzene because some iodine. This is because of this brown color. So that means the compound which we have prepared, this was making the organic gel. So it has the additional value I will be talking about in the following slides. What is the additional value of this gelation? Importantly, this molecule, when was subjected for the gelation with the aqueous solvent or polar solvent, because these solvents are non-polar like benzene, toluene and all. But here if you see the DMSO and water, like 1:2 ratio, that means 2 excess of water, or even ethanol and water, like 1:2 ratio, that more water and less organic solvents, this was perfectly making the gel. So this we can say this is the molecule which having the property of organogelation as well as the it's making the hydrogel. So this was the preliminary study which we have performed, where we identified, oh, molecule is perfectly fine for the gelation of organic solvent. And this is the detailed studies, like in how much time generally it takes to make the gel. So at room temperature with the benzene, it takes 9 minutes, almost 9 minutes and 54 seconds. But if you do the same gelation into the ice bath, then it takes 4 minutes. So almost half of the time what is taking at the room temperature. Importantly, what one interesting thing we noticed about this molecule, that it makes the sonogel. What is the sonogel? Generally, the sonication is a property, and the sonication where we use the uh, energy, like to dissolve anything. Okay, we used to have the sonicator in our laboratory where we try to clean out our stuff or we try to dissolve anything. But this molecule was interestingly making the sonogel, means gel formation under sonication. So within 6 seconds, you see, this molecule with benzene, within 6 seconds it making the gel very quickly. So we have studied like room temperature, ice bath, and even the sonication. Timed variation was there, like in room temperature 9 minutes, ice bath 4 minutes, but under sonication within 6 seconds. And this was very nice TG. TG means transparent gel. So if you see with other different solvents like toluene, xylene, metaxylene, paraxylene, this is making perfectly fine gel. Even this ice bath 10 seconds. Metaxylene was gelated within 10 seconds. Whereas under sonication, only 2 seconds. 2 seconds means just you take the molecule, put xylene, and just even dip inside the sonicator. Quickly it makes the gel. So this is how we measured it by using the uh, this watch, like which you can measure even the fraction of second. So within 2 seconds it makes the gel. And without other solvents, see, I'm not going to read out all these things. You can easily see here. So like in mesitylene, 45 second, within 45 seconds it makes the gel at room temperature, but in the sonication condition only 3 seconds. Idobenzene, okay. Under normal condition, room temperature 9 minutes 30 seconds. Average at ice bath, it makes 2 minutes. It takes 2 minutes, and with sonication only 2 seconds. Perfectly fine. So once another property of this material, like when we have made the gel, so we have to find out how stable that gel is. Like let's say if it makes the gel, but if gel is something like holding the solvent molecule. But if that binding is not strong, if the solvent molecule is not properly holded, what will happen? After some time, they will get dissociate. So the stability of gel is very important. So we have studied the stability of this gel by using the rheology. So you see here, so our gel, like with the toluene or hydrogel, okay. Uh, so this is quite stable, the toluene gel, because this, if this is going a straight line, that means this is the stability. If this is coming down like this, that means it's not stable. So this was quite stable gel. And whereas the G dash value with the toluene gel was also quite good, you see, as well uh, as this with the hydrogel. You see, hydrogel was also quite stable, straight line. So what I'm trying to convince you that the gel which is formed by organic solvent like using toluene or benzene or mesitylene, or even the gel which is formed by the DMSO water or ethanol water like hydrogel, they were quite stable, and their solid-like behavior and liquid-like behavior, they were very much constant with the time. So that was quite good results. So next, once you make any material, so we had to study there how does it actually got assembled, what's the whether this is thread-like structure, whether this has some network. So we have done the SEM, we have taken the SEM images of the gel. So these are the SEM gel of toluene gel, you see. So this is like very thin thread-like structures. But when we have taken the same images of hydrogel, you see, this is bit different than what was the with the organic gel. So this is network kind of structures is forming. So that what does it mean? It means that the organic molecule along with the solvents, they were making a very well defined 3D structures. They are holding the solvent molecules in a very well oriented form. So this was all about the study of studying the property of that gel which we have prepared by using that molecule, carbohydrate based molecule. Now next was whether, because the oil spillage is a big problem, uh, we have learned like 3 or 4 year or even 5 year back in the Mexicans this Gulf, there was one ship which was carrying the oil. So that was shrunken and lot of oil was floating onto the sea level. So there are methods by which we can collect, or we can collect that oils, because oil is lighter than water, so it comes onto the surface. So we can scoop it out, or we can simply uh, actually there are methods by which we can remove that water. But one another way of removing that water is through the gelation, because if you have the molecule which are phase selective gelator. Phase selective gelator means like you have water and you have oil, and oil is lighter than water, so it comes onto the surface. And if you have molecule which you can simply spray on it, and if your molecule selectively making gel with the oil, so you can simply take out that oil along in the form of gel. Water can remain there in the pure form. So that is called phase selective gelators. Like out of two phases, only one phase is got gelated. So we have studied the phase selective gelation property of this molecule by in laboratory by just taking the petrol. So you see this first slide A here. There's a water, and you see this little brownish color. This is a petrol. So we have well mixed up and then just kept it. What happened? All petrol came up, and then we have applied our molecule little bit onto the surface. What happens? Like after a few minutes, the petrol which was on the surface, this was got completely gelated. You see, this same oil when got inverted, so this petrol here in the gel form is enough strong that is not allowing the water from here, because this is liquid, come down. Then we have scoop it out, this petrol in the form of gel, you see in the which is placed onto the petri dish. Not only petrol, we have studied the gelation property with the diesel as well, because diesel is a bit heavier petroleum products, and the petrol is lighter than diesel, because diesel has even bigger hydrocarbons, so therefore it is denser as well as heavier. So gelation property of diesel was also studied, and we demonstrated that through the phase selective gelation, either petrol or diesel can be gelated. Now once you have this kind of gel in the little larger amount, which actually we did, you can simply what you do, then simply take in round bottom flask and heat. Once you heat, what will happen? The thermal heating will break this gelation, because it provides the energy. It's what is holding get poor, and slowly this get dissociated. So what will happen? The gel and means your molecule will be separated, and the petrol will be separated. then you simply distill out the petrol, and then what will happen? Only your molecule in the solid form, because your molecule is solid, will remain into the round bottom flask. Then you can reutilize for the gelation of next batch. So that means this is the way we can separate the petrol or diesel through the phase selective manner by using this material, and which can again the molecule can reutilize for the gelation of next batches. So this was quite interesting result. So we demonstrated that the petroleum oil, diesel, this they can be removed from the water molecule. Another thing which we have performed actually to study whether this molecule is helpful in making the nanoparticles, because making the nanoparticles, this is one area of research, and the challenge which actually happens with the preparation of nanoparticle is to get the uh, nanoparticles of the same size, okay, like homogeneous nanoparticles. Because when we do the nanoparticle synthesis, the lot very different size of mixture of nanoparticles you get. Let's say someone is 30 nanometer, 50 nanometer, 100 nanometer, or even some is very smaller like 5 nanometer. So you get all different size of nanoparticles when you do the conventional synthesis of nanoparticle. So making the homogeneous nanoparticle is a big challenge. Still, there are methods now people have been actually has been developed, but it's still like in the say they like making the nanoparticle of the desired size in the homogeneous form is still a challenge. It's still an area where people are working. So we thought, uh, because this molecule has the gelation property, can we make nanoparticle in the presence of this molecule? Then what will happen? Because this make the gel. So gel is, I said this is very stable structures where molecule are present in the predefined form. And if this is also able to make the nanoparticle, are able to reduce the, let's say, gold or silver, so maybe when the nanoparticle will form inside the gel, so they may have the similar size, okay, not much. They may makes the homogeneous size of nanoparticle, and those nanoparticles will be trapped inside the gel, and then therefore they won't be making the aggregates. Another difficulty of making nanoparticles, because once they are in a nano form, they try to aggregate together, so they make the clusters. So those kind of things can be avoided if I try to make the nanoparticle by using these organic molecule which have the gelation property. So we have took the AuCl3 in the phosphate buffer, and we found that yes, because these are very specific colors. This AuCl3 is colorless. Once this started making the nanoparticles, you start a light blue color. Light blue color appearance indicates that the gold now got converted into, AuCl3 got reduced into Au, and that Au is present into the nanoparticle size. Similarly, with the silver nitrate, because silver nitrate when get reduced to the silver, and when this is present in the nanoparticle size, it makes very definite color of, this color, like golden light gold colors. So the first thing what we have noticed, that these molecules, they are able to reduce the AuCl3, or they're able to reduce the silver nitrate, and that's Au or silver makes the nanoparticles in the form of nanoparticles. This is the first proof of concept. But the second was like the one which we were interested more, because making gold particle or silver nanoparticle is not big challenge, because there are many methods by which we can make it. the thing which we were more interested, whether these nanoparticles are the homogeneous size. Okay, so see here. So this is the actually this is the TEM images of the nanoparticles which we have prepared. If you see, this A1 is the, sorry, so this you see here, this is the zoomed structure, and if you see most of the nanoparticle, their size is almost same, although there is some small dots you can also see, but majority of the nanoparticles, they were of the same size. So that means in homogenized form. Similar was true for the silver as well. And we have studied the UV visible spectra of the nanoparticles because they are very specific physical properties of these nanoparticles. So you see this clear bands were seen. So that is helpful in the characterization that the now silver is in the silver nanoparticle form. So this was, that means these compounds, the compound number 3 which we have prepared, which we have synthesized, it has the, this material has the value for making the hydrogel and organic gel, as well as phase, it we have like a phase selective gelators, and also it makes the homogenized, or it makes the nanoparticle of the homogeneous form. The last application of this molecule, we plan to see whether this gel, this organic molecule, having the property to remove the dyes, because the water pollution or the pollution which is coming from industry like dyes is a big problem, the big challenge, and how to remove this waterborne pollutant, or these dyes which is what simply drain into the river. Here in Delhi, you see many industries, and these colorful dyes through the canals, they goes to Yamuna. So can we treat that water which is coming from the industry, especially this the industry which is involved into coloring of the this jeans pant, and this many industry which is the dye is a color. Hmm. So we found that the molecule, that's our compound number 3. See, this is rhodamine dye. This is pink in reddish in color. So when we applied our carbohydrate gel molecule, like gelator molecule, to this dye, and after 24 hours, you see almost all of the dye color has been gone. So now you have almost pure water. As if you see this versus this, so this is, you can think about a dye as a pollutant in the water, and here all the pollutant has gone. This has been taken trapped by the gel, and this water is almost pure. And we studied the how the this dye is getting gelated with the material which we have prepared with the time by using the UV absorption spectrometry. So in the beginning you see this red color, this is the absorption of this dye, so it's very strong absorption around 550 nanometer, which is very characteristic for this rhodamine molecule. And at 0 hours it's quite high, and with the time, like after 1 hour, after 2 hour, after 3 hour, after 4 hour, you see almost 50% of dye has been gelated by the that material. And slowly with the time, different time intervals, like even after 24 hours, you see this is coming as almost a straight line. That means there is no dye, because if there is dye, then it must show the absorption. So that means within 24 hours we can remove. So in terms of industrial application, how we can think about it, let's say there's an industry which produces the water pollutant, and if by collecting that water pollution, pollutant with the dye in a tank, and then applying this molecule and leaving it for 24 hours. After 24 hours, you can remove this gel because it is absorbs, it is having the dye, and water you can simply throw into the canal which may go to the river. So this is better purified water. And this gel, uh, you can remove it, this dye from the gel, and you can reutilize the gel, or you can process it, this gel, in different way to separate the organic molecule which you have applied, carbohydrate based, and the dye you can reutilize for coloring. you know this is we thought about the industrial application. Another, so this was one, this is how we completed this project. So we, how much time do we have? So this is 10:56. I think I have another 20-25 minutes, isn't it, Bindu? Hello,
Yes, I think, yes.
is Bindu is there? I think she got disconnected.
Yeah, I think you can go until 11:30, I guess.
Okay, okay, thank you.
Sure.
So this was the one story, and this got published. Another project in this line, what we have started, and now that is also completed, got published in Langmore 2019, like the abstract which I shared. So this reference is there. So in this case, in this particular project, what we have planned to introduce a triazole ring into the carbohydrate molecule. So this, let's say R1 and R2, this could be different carbohydrates, and these two carbohydrates molecule may be linked with the triazole ring. And triazole ring, we all know that can easily be prepared by there are many means, and one very common means or method for making the triazole is click chemistry. If you have alkyne and if you have azide, and by using the copper as a catalyst, you can do the click chemistry, and you can get 1,5 that substituted triazole ring. Sorry, 1,4 substituted one. And whereas another, if you use the rhodium, then you can get 1,5. So the beauty of the triazole ring is you have 3 nitrogens, and there is a 1 hydrogen here, which is acidic in nature. So this hydrogen here can act as a hydrogen bond donor, whereas a nitrogen 2 and nitrogen 3, this is a potential hydrogen bond acceptor. Okay, so whatever donor and acceptor present into the carbohydrate molecule, like with R2 and R1, but you have additional hydrogen bond donor and acceptor into the triazole ring. So maybe if you incorporate these triazole rings in between the carbohydrate molecules, you may have a better material property. Along with this triazole here, it is aromatic in nature. So triazole ring is aromatic in nature. So this may have the pi stacking properties. So the three additional value. One is hydrogen bond donor, one is acceptor, and then pi stacking. So by considering that property in mind, I have designed these 5 molecules. Okay, 1, 2, 3, 4, and 5. And so actually, these are the molecules which actually reported earlier in different papers like Ingavanti 2016 and Chemical Communication 2014, or even Langmuir in 2011. So these kind of molecules were already been prepared, where what they have done, they have put a carbohydrate unit, and they introduced the triazole, and there is an alkyl chain or aryl group here, or any other sugar moiety here, or long chain alkyl groups, and they observed that these molecules, they have very good gelation properties, and they makes a very strong gel which is stable even for many many days. So that means there are literature reports where the triazole ring has been used for the gelation purpose. So we have designed this molecule. You see, this is 6 molecule here, where what, and if you see these molecules, in most of the cases, not most, in all of the cases, the triazole ring was introduced at the carbon 1. So this is called carbon 1 in carbon compound. So, or you can say anomeric position. The carbon 1 in carbohydrates also called anomeric position. So, so these all reported molecules, they have the triazole ring at the carbon 1. What we have planned to see the effect if the triazole ring at the position 6. So this is my molecule. This is carbon 1, this is 2, this is 3, 4, 5, and 6. So we plan to make the molecules, these 6 molecules, where we have the triazole ring at position 6 rather at position 1. And what we also further actually designing element, the triazole followed by a phenyl ring, triazole followed by a amide bond. Okay, amide bond with the alkyl chain, amide bond with the simply aromatic chain, or simply aliphatic chain, you see CH2, CH2, CH3, or an alcohol. So we have introduced the structural diversity in this molecule, and this is the synthesis how we have prepared. And again, starting with the N-acetyl glucosamine, glycosylation followed by tosylation, we got compound number 2. So here, what we have done, we simply treated this molecule with the sodium azide at 80 degrees centigrade 6 hours. What happens? The azide replaces the tosyl, and we got azido derivative. Now we have done the benzylation here by using sodium hydride. So we got benzylated product with the azide group at position 6. Now we have used different alkynes like phenyl acetylene or amide containing acetylene, and under microwave condition within like 5 minutes we have at 20 degrees centigrade by using copper sulfate, we exclusively got this product in 99% yield. If you do the same reaction under room temperature condition, still you get the triazole ring, but it takes around about 24 hours. Okay. So anyway, by using either thermal or microwave, we were able to make the triazole based compound 5A, and similarly other molecules have been prepared by using corresponding acetylene. When you use phenyl acetylene, you get this molecule. But when you use paramethoxyphenyl acetylene, you will get this one. Okay, so this is how all molecules got prepared through this route. Once these molecules were prepared, we have started looking their material property in terms of gel formation. You see the compound number, this, where the phenylacetylene derivative of this triazole compound. This was making gel. G means gel formation, S means soluble, and P means precipitate, means insoluble. So this particular compound, it making gel with the toluene, with the xylene, with mesitylene, xylene and mesitylene. But when it comes to the halogenated hydrocarbons, this was not making jet at all. It's only making the compound was getting completely soluble, no gel formation. If you remember my previous slides, where we had a molecule which was making the gel even with the halogenated hydrocarbons, whereas compound number this one where the free hydroxy group was present, this is the compound number, if I say this is 5E, so this one is not making gel with any of the organic solvents. And this amide one, this got all precipitated. This is not soluble actually. And this is the one which has the alkyl chain that was also soluble. That means out of these 4 or 5 compounds which we have designed and prepared, only the compound number 5A, this was having some hope because it was making gel with the hydrocarbons. And interestingly, this molecule, which was not making the gel with the halogenated hydrocarbons, but it's making perfectly fine gel with the diesel, with the petrol, and with the crude oil. So this result was very much exciting because may, finding a molecule which makes the gel with the crude oil is something better, like something something exciting, because the crude oil separation from water is still a big challenge, because petrol is something purified form, diesel is something purified form with the higher hydrocarbons. The crude oil, it has everything: diesel, petrol, even solid hydrocarbons. So making gel with the crude oil is something was exciting. And this compound number 5A, it was having this property. And this is again the time, how much like time does it takes to make the gel. So like compound number 5A with the benzene, it's making transparent gel, and with the mesitryl also making the transparent gel. And time generally takes like 10 minutes at room temperature and 3 minutes at the room temperature. But when you do the sonication, so it makes the gel like within a 20 to 25 seconds, within 25 to 30 seconds. And these are the pictures of the gel which has been found by this molecule. See, very nice transparent gel. It's not coming out when the when these vials got inverted. This is the with benzene. This is the toluene. But this compound does not mix the gel with the halogenated hydrocarbons. Like a small change, it changes the total property of the molecule. And now, because this was my second project in the similar line, so when we submitted this like whole paper for the publication to the Legmoor, which is a ACS publication, the referee asked can you, because saying something in science is easy, but proving something is difficult. So they asked, can you give me some experimental proof that the gel formation which is taking place here is where it's through the hydrogen bonding. Okay, so then we thought about some experiment, because one way of proving whether there is a hydrogen bonding or not, we can do through the IR spectroscopy if we have very predefined specific bands for that particular hydrogen which you thought about the participation of the hydrogen bonding. Because when you dilute the sample, if this is intermolecular hydrogen bonding, that bands will shift. But we designed we thought about even something more precise experiment, which was the proving the hydrogen bonding through the NMR spectroscopy. And these are actually results. So once the gel is formed, like the compound number 5A, when the NMR spectrum of this compound was taken in DMSO, so this is the hydrogen of the triazole. Triazole, when I say this one, this hydrogen here, this here, okay. Rather I go better. This hydrogen, okay. So this is the triazole hydrogen. So in an NMR spectrum of compound 5A, so this was appearing around here, around 8.6 ppm at 25 degrees centigrade. When the temperature has risen up from 25 to 30, from 40 to 50 to 60, you see there is a clear shift from 8.6. So this particular hydrogen are shifting upfield. So that indicates that this triazole hydrogen, which was a room temperature, even low temperature, this is got shifted downward, but high temperature, this is shifting up. So this indicates now this hydrogen is got freed from some interactions at higher temperature. Therefore, it's got shielded, and therefore if it is got shielded, then therefore upfield is taking place. Another very important shift which you can see was observed for this particular hydrogen. You see this doublet. Once the temperature got risen up, so slowly this doublet got shifted towards this doublet, and this doublet is not shifting. This is at its own place. Now this one is got shifted and got merged with this one. Okay, so these shifting of the triazole hydrogens is around delta max, I would say around 0.117 ppm. Although this is into the points like 117 shifting, but in proton NMR spectroscopy, this shifting is quite remarkable and it actually matters. And another one, the NH of the triazole, like free NH, this got shifted almost, how much? Like from 8.15 ppm to it gone up to 7.85. So this is around 0.16 ppm of field shift. This clearly indicates that this particular molecule 5A, which having the triazole ring and the triazole hydrogens participates into the hydrogen bonding. Once we have this molecule, once we have this gel, as I said earlier, their stability is very important. So you see, this is the picture which shows the through the rheology data that this molecule, whatever the gel is found, is quite stable. And this is the same images. Now these in here is even better. So this bottom one is actually gel with a toluene. You see, very like this is very nice network thread-like structures. And whereas the same compound, when you make the mesitylene gel, its arrangement is a bit different than what is with the toluene. Just like changing one small because chemically the strictly difference between mesitylene and toluene is only one extra methyl group. Okay, so you see the orientation of this molecule with this or these organic solvents is quite different. Okay, thread-like structures. Now, as I mentioned earlier, this molecule was having the gel property not only with the petrol and diesel, but also with the crude oil. So we have collected the crude oil from this from refinery, like from this petroleum industry, and then we have mixed this crude oil with the water, and then we applied our molecule. You see, whatever this oil was there floating on the water, this makes quite nice gel, and this gel you can scoop out and put into the spatula. Put into the this petri dish by using the spatula. So these are the different experiments where we have shown that different API grade crude oil, because the crude oil have different grade. So like Kuwait crude oil or Iraq crude oil, they're different name of they have a different grade of crude oil. Like the place from where this has been extracted out, so that shows its different API grade. So this compound 5A was making gel with most of the API grade crude oil, and their gelation property happens very nicely. And these are the rheology data of that crude oil, which actually shows this was quite a stable straight line. Okay. Now, as I did with the first molecule, we also did similar experiment, and with this 5A molecule, see here, even the earlier molecule was a bit better. So this is the rhodamine dye which is soluble in water, this color. When the gel is applied, so you see this is the gel which is attached to the wall. What happens? This absorbs all the dyes, and water almost become colorless. And but this molecule 5A was not able to quench or not able to absorb the methyl orange dye. You see, this is the methyl orange dyes, and here the compound is applied, but still that dye remain in water. See, it's not even after 11 hours, this dye is not getting absorbed by the this gel molecule. That means like this molecule is selective towards rhodamine dye and also crystalline violet. Okay, this is crystalline violet dye, and when you apply this compound, you see the dye is completely got absorbed into the gel, and water remain colorless. See, there is no absorption after 10 hours, okay, 10 and a half hours. So that means the compound 5A, it was able to remove the dye from the water, that was the crystalline violet and rhodamine. But methyl orange, this is not get absorbed by the this particular compound 5A. That means it has some selectivity. So in conclusion, I can say we have synthesized a multifunctional carbohydrate gelator, which was in previous project compound 3A, and which was showing the gelation property with the organic molecules, with the organogel, as well as it was making the hydrogel. And in another project, we have made the triazole-based gelators as a material, which was able to make the phase selective gelation with the petrol, with the diesel, as well as the crude oil, which was the most exciting. The earlier molecule 3, which was prepared, it was making the it was studied for making the nanoparticles with the silver and gold. We observed that using these molecules inside the gel, they make the nanoparticle which are homogeneous in nature. And the gel formation with these compounds, even they can be achieved within 2 seconds to 5 seconds by using this indicator. And these organogelators, compound 5A or compound 3, they were efficiently removes the various water-borne synthetic dye in a specific rhodamine and crystalline blue. Okay, with this, I think I should conclude here. So, and I'm thankful to Banaras Hindu University and also my students Chintam, Narayana, and Priti Kumari for carrying out this work. And I'm thankful to you all for your kind attention. And this both the topics which I have presented here, they have already been published. One in 2017, other in 2019, and Langamore at 2019. And those of you who are interested into the details of these compounds, much deeper detail, you can go through these papers. A lot more analysis and studies has been compiled there as a supporting information. Thank you very much for your kind attention. And if you have any question, I am happy to answer.
Thank you, sir. Thank you very much for your this nice presentation and quite informative and interesting information, sir. And you have invited students also that if they want to come with you for understanding these kind of reactions or research, they can join you. So I think that's a very welcome gesture, sir. And hope we like young teachers and students, they will be benefited with such kind of research, and they will definitely come in contact with you if they want something deep in this area. So now I invite questions or queries from audience if they have any. If anybody has any query, then they can unmute themselves and can connect. or they can raise their hands. I think you explained very well, sir. So they are not having any query. So I want to thank you, sir, from the Department of Chemistry and the associated organizers, Vigyan Bharati Vibha and American Chemical Society, for giving this nice talk on this 6-day online short-term course on advanced materials for Aatmanirbhar Bharat. And yes, during this prevailing uh, pandemic, if we are able to develop different kind of molecules or smart materials, then of course we will be progressing ahead with the mission to achieve this Aatmanirbhar Bharat, which is a big vision of our honorable Prime Minister. So, and to add to this, I would like to share with you, sir, that our university has developed a central research facility where we have procured a number of instruments for the characterization of these smart molecules and materials. Like we have procured SAM, we have got FTIR, AFM, and in fact, all the participants who want to avail these facilities at the university, they are more than welcome, and they can go through the proper procedure to avail these characterization techniques. So, sir, we really need speakers like you and researchers like you who can help us out and who can help our students to go deep into the research. Thank you so much, sir, for your valuable time and this valuable talk. Thank you so much.
Okay, thank you, Bindu. Thank you very much.
Now we have with us our next speaker, Dr. Chabda, Dr. Arvind Chabda we have, and he has connected. Dr. Arvind, are you there?
Yes, go ahead.
Yes, yes, sir. So now I take this opportunity to introduce our next speaker, Dr. Arvind. Dr. Chabda did his master's in biotechnology from Guru Nanak Dev University, Amritsar, and PhD in biotechnology from CDRI, Central Drug Research Institute, Lucknow. After completing his PhD degree, Dr. Chabda joined University of Connecticut Health Center, Farmington, Connecticut, as a postdoc fellow. Dr. Chabda served as an assistant professor of medicine in the Department of Medicine and in the Stem Cell Institute at UCHC. Dr. Chabda joined Amity University, Haryana as a professor and director of a Stem Cell Institute. He has a cross-disciplinary research expertise in the field of human cancer immunology and immunotherapy, human pluripotent stem cell research, molecular biology, and nanotechnology. He has published more than 30 manuscripts in prominent international peer-reviewed journals. His research work has been widely cited and has earned him award and accolades at international platform, including Overseas Fellow of Royal Society of Medicine membership. Dr. Chabda has also served as an editor and as editorial board member for several prominent research journals, including Scientific Reports, Oncotargets, and Therapy and Stem Cell Discovery Journal. In addition, he also served as a peer reviewer for several reputed international research journals, including Journal of Immunology, Journal of Leukocyte Biology, Vaccine, and Expert Opinion on Therapeutic Targets. I welcome you, sir, on behalf of Department of Chemistry and Vigyan Bharati Vibha, American Chemical Society, on this platform of 6-day online short-term course on advanced materials for Aatmanirbhar Bharat. You have very well planned your title, that development of indigenous technologies and materials for biomedical applications for an Aatmanirbhar Bharat. Sir, in this prevailing pandemic, we really want to develop, like for last 1 year, the whole world was struggling for the vaccine for this pandemic. And if we can add something to it for biomedical application, and that too in this prevailing pandemic, I think that will be a boon for the mankind. I welcome you, sir. Now stage is yours, sir. Your slide is quite visible.
Sure. Thank you very much, Dr. Mangla, for inviting me. I thank the organizing committee for making me part of this exciting conference. And thank you very much for your kind introduction. So since we have a lot of students, and I think the theme of this meeting is how we can make Aatmanirbhar Bharat and develop different technologies. I will start by saying that I'm no way like pure material scientist. So as you have already read my little bit of research expertise, I have cross-disciplinary expertise, and we have material scientists in our group. So I would like to start by acknowledging Dr. Monica Vass, who is also part of this gathering today. So she is a very excellent supporter that I've gotten and collaborator. So since we have a lot of students over there, so I thought like I'll start like by exciting them, like what do we need to build an Aatmanirbhar Bharat, and in that context, like students should be aware of what is intellectual property. If you're developing something, if you're doing some research, doing research is not the only thing. You have to protect your intellectual property also. Because like if you can develop a product and somebody else is going to reaping the benefit, so then you are losing money. Science is doing science also for the sake of science, but there is a lot of money involved in the science. So students should be aware of that. I think when we go in as life sciences or other chemical sciences field, students sometimes feel that we are less fortunate in terms of monetary benefit. So I would like to start by saying that there is no dearth of monetary benefit in these fields, but you will have to acquire knowledge, and then you have to acquire traits, how to convert that knowledge into money. So I will start by telling something about intellectual property, and then we will go to, like, since we have Science Day coming up, so we will start by okay, why it's not going to the next one. Okay, so then we will be talking a little bit about science in terms of what is different approaches by which you can treat cancer, and what are stem cells, and feel assured, I will not be belaboring you like too much with the intricate details of these subjects, but like just enough so that like it can enthuse your imagination. Because like whatever field these days we are doing, whether it's a biology or anything, like at the heart of it is a chemistry. So there is no DNA without chemistry bond formation, and there is no life. So I'm just a bunch of biomolecules sitting together like in a good architectural manner, like put together by the nature. So we will be talking some of the scientific aspects in these fields. And then in the end, as we discussed, like we will be talking about some of the technologies that we have developed and that are very relevant in context to the COVID scenario that we are going through, and how we have developed it and what are their applications. So we'll be ending with that. Like so to start with, like as I said, like what is intellectual property? So generally, as you describe, like any property, it can be a tangible property like your house or anything like that. But like intellectual property is like intangible property. So this is not a physical form, but nevertheless, it is similar to other legal property rights that can be sold, that can be leased, that can be gifted, that can be licensed or waived, and allow the holder exclusive right to use or commercially exploit the product of his or her intellect. So if you have made an innovation, then you are free to make a product that we will be seeing some of our product that we have built. You are free to sell on your own, or you are free to license them to a company which will give you some royalty on that. So this is basically intellectual property that you can make money out of it. So you are sitting there and then you get an idea. So, and then how do you protect that idea? So it can be different forms of intellectual property. Like one form is a patent, but there are lots of other kinds of intellectual properties over there, trademark. So you're going through a highway or anywhere, you see a McDonald's sign, and you recognize there is a McDonald's restaurant over there. So that sign that shows you that there is a McDonald's over there, that is a intellectual property or a trademark for that particular company. You are going in a car, and many car passes through, and you can, without knowing the person, you know, like it was a Tata, it was a Toyota, it was a Hyundai, because just by seeing the trademark of that company. So these are all intellectual properties, and that's how like different companies protect their intellectual properties. You read journals, you read books, and they come under this another type of a copyright property, intellectual property, so that nobody can reproduce those products without your permission. So, and you can get a royalty out of that. You write a book, you get a royalty out of that because you have a copyright on that content you have developed. Then, of course, patents. So we will be talking a little bit more in detail about the patents and the process about it. And then mechanical designs, trade secrets, these are other forms like so, but constitute a particular invention to be patentable. uh, so the reason why I'm telling you these guys is because like you have to whenever you are doing some taking a research project or anything, then you have to think whether there is a good enough chance to get you a patent out of that science. And the work that we have recently developed, and it has been accepted also for publication. I'm pleased to say like that there is a master's students in our chemistry department, like who did the whole work. And then she got a patent out of that in her name, and she got a publication also in her name now in the master's level itself. So this is how you should think about your research project design and everything. So what are the key requirements for a successful patent application? The product or a means invention that you have created, it has to be new. Means it has not been done before, because if it has been done before, then there is no novelty. So there has to be a novelty feature in there. And that novelty is determined whether you have introduced a inventive step. You have some inventive step there that makes your invention unique. And if it comes to me, you have invented something and I'm from your same field, and then I find that there is no major change in the reaction that you have done or a product that you have designed. For example, a shape of a bottle, you just make it a curved form or something, so it may not be qualifying enough for patent, maybe for a design but not for a patent. So non-obviousness is another criteria, and it has to have some application attached to it, so that this can be applied for use or something. So these are the key requirements based on which you can your invention can qualify for a patent. So in terms of India's means intellectual property scenario, as our GDP is rising, so is rising the different types of intellectual property right filing. And not only the filings, but these are the filings again by resident, non-resident, and Indians living abroad, in terms of that means color coding and the carded number. So we are making progress substantially, but like you have to always feel content in what you are doing, but then you have to look into like a holistic scenario also. Where do you stand in terms of a global rating? And when you talk about global rating, we are a country of 1.3 billion people. So if you see this figure, means this figure was encouraging, but if you compare it over here, we stand nowhere near to the top patent filer, which is China. In 2018, they had 1.5 million patents, while India was just 50,000 patents. So government is making a great push to make Aatmanirbhar Bharat startup culture to promote that. and encouraging young people like you to think innovatively and then develop innovative solutions to social problems and file more properties, intellectual properties, patents, and set up some startup companies. So there is nobody holding you back, and there is no dearth of the imagination that you can think of, but you have to acquire knowledge for that. So what else that this intellectual property creation brings to you? If you can get a patent in the master's level, this will help you get a good PhD opportunity, career opportunity. It will bolster your CV. Entrepreneurship, so you will have created an intangible asset in your name. And if you can license the technology, you can fund your studies also. So that's what it is. And then of course, like as you grow in your career, so there will be lots of other opportunities that will come along with it. It holds true for all the faculty also. So in our career growth through the course and our promotional scenario. So this was just a primer to enthuse the students about what are the different ways that you can design your research and then protect your ideas and reap the benefit, monetary benefit out of those ideas. We'll be sharing some of the patented technology that we have developed towards the end. But like, let me start the some scientific work that we have done in this cancer immunotherapy field and the stem cell field now. So in this, when we talk about engineering, like we can always think about engineering. Engineer is nothing but a somebody who can engineer a machine or something. But like I'm gonna tell you, you can engineer your body also. So we'll be talking about like how to engineer a immune response. And since we are living in a scenario of COVID, so these days lots of efforts are going on how we can engineer our immune system to fight effectively against COVID virus. And the technology that I'll be sharing with you, that holds true for COVID also. So that I will correlate, but like example that I've taken is on cancer, which is a big challenge that is developing in India also. So how does the cancer develop? I think these days, lots of us within our immediate family or in a friend circle know somebody who has acquired cancer or who knows somebody in relation who has acquired cancer and potentially lost the battle also. So the cancer develops like in our inside our body itself, unlike COVID, which is a virus which comes from outside. So if a intruder is coming in your gathering, then you know like that this is a foreign element, and you can either make it part of your group or you can eliminate it. So our immune system recognizes any foreign element and fight it off by means of different immune reactions. But since cancer is developing inside our body, and how it develops by accumulation of the mutations. So this is harder to eliminate. So we will talk about what are the different ways this is harder. But like to start with, a number of mutations get created and some cells become cancerous. They secrete some molecules which you can together name as danger signals. So these danger signals get recognized by our immune system, and our immune cells come and detect them and fight them off. So this is the reason why majority of us do not develop cancer during the most of the early stage of our life. This cancer cells are killed and the healthy cells are left behind. But some cells in certain individuals, these cancer cells can escape and they keep on accumulating more mutations. What are mutations? So essentially, like as I was saying, like we are nothing but a bunch of different biomolecules. And in our case, our cells are made up of cell membrane. Then you have a nucleus, and within the nucleus, there is a DNA which is packaged there. And then within the DNA, there are open reading frames which encode different proteins. And that's how like we function, our body functions. So if there is a change in the sequence of that particular DNA, then the function of a protein is lost. So the normal function is lost, a protein becomes abnormal. So, and if there is a mutation in a essential gene, then that cell becomes cancerous. So this is what is the accumulation of mutations. And once a threshold of mutation is achieved, then these cancerous cells, they become metastatic. What do we mean by metastasis? That means if a cancer is developed at one location, then and that particular cancer acquires metastatic ability, then it can move to different sites of the body and then metastasize there or grow there. And that becomes a harder to cure phenomena. And they secrete certain molecule by which immune system cannot even fight that. And then that patient can succumb to the disease. So what are the different therapeutic approaches that are available? So after surgery, once a cancer has been removed, patients are put on different chemotherapeutic drugs or radiotherapeutic drugs by which most of the highly dividing cells. So because like cancer cells are dividing highly, means in a unregulated fashion, because they have lost control over their uh, cell death and cell survival mechanisms. They just keep on growing. Usually in our body, all the cells have a finite life cycle, and they get replaced at a certain time. We will talk about it a little bit more in detail when we talk about stem cells part a little bit later on. So since they are highly dividing, so these drugs, they target the cells which are of highly dividing nature. So they detect those cells and then they kill them specifically. But since other cells are also dividing, not so fast, so there is a side effect associated with these approaches. So there is another approach that has been developed recently, which is immunotherapy, where your immune system is programmed to fight these diseases, whether it's a cancer, whether it's a COVID, and this can be antibody-based therapeutics or T-cell-based therapeutics. So you can design those therapies. And that way, since we can get vaccine as we are born, vaccine against the polio, vaccine against tuberculosis, so we don't get any side effect associated with that particular vaccine. As we are knowing with the COVID also, millions of people have taken vaccines for COVID, but there is very small minuscule function, means fraction, which has gotten any side effect and which is also manageable. So because you are just programming your immune system, and since that is a part of our body, so there is no side effect associated with. So these approaches are being tried to boost the immunity against cancer also. So, and this holds true again for COVID, and this vaccine against COVID will also work in the similar way. So there are three key cells which are essential for fighting any disease, which is first is the antigen presenting cells. This is kind of a like your teacher, which will take up the knowledge from different sources, process it, and present it to you. So this antigen presenting cells takes different type of antigens from different pathogens and process through MSC class 1 and MSC class route. MSC class 1 goes to the CD8 positive T cells, which are mainly your killer effectors. They are like an army, which will identify the target and kill it. Then CD4 cells, any antigen that comes from outside, that will be recognized by the CD4 T cells, and they can either help this army or they can put a break on this army. How does a cancer moves or escapes our immune onslaught? So there are different mechanisms by which cancer escapes. One is these cancer cells, as they are growing, they downregulate the expression of these MSC molecules and also certain molecules by which these tumorous properties are gained by these tumor cells. So these cells lose those antigen or those MSC molecules. So immune cells, to immune cells, they become invisible. So that's how like your immune system cannot fight. Another, they express these FAS and other trail and other ligands. So these are death receptor molecules. So these T cells, when they get activated, they also express the receptor for these molecules. So the idea is these tumor cells can essentially kill these T cells. So this is known as a tumor counterattack hypothesis. So instead of killing the tumor, tumor is essentially killing the T cells in a sense. So that's another mechanism by which these tumor cells fight off. And of course, another mechanism is immunosuppressive microenvironment. What do we mean by that? So if we take an analogy of a car, immune system is a car and there is a acceleration there is a brake there. So if there is a acceleration available, then the car will move further. But if there is a brake that has been put in place, then that car cannot go further. So these are some of the mechanisms which serve as a brake. So macrophages, dendritic cells, Th2, these are some of the cells which gets skewed towards immunosuppressive microenvironment, and these are the effector molecule, interleukin 10, TGF beta. These are molecules that are secreted by these effector molecules, which impart these negative function. And that's how the effector mechanisms gets activated or down modulated. And these means immune cells cannot kill the tumor cells. How a vaccine work? How does a vaccine is created? So basically, like if the individual has gotten cancer or if it is gotten a means COVID also, so you take either these antigen presenting cells, and they are technically known as dendritic cells. So you take these dendritic cells and then you express this disease specific antigen or a gene, and then introduce these cells back into the patient. Or you can take those antigenic small peptides, as I was saying, MSC class 1, class 2 molecule. Those small peptides you can just take, chemically synthesize them, and then pulse them and then use them as a vaccine candidate. Or tumor cells also you can modify and then use them as a vaccine. In case of COVID, we also know like there is a spike protein. So you can take a spike protein, express it in a dendritic cell. This become a spike protein based dendritic cell vaccine for COVID. So this is how, and then since these vaccine will go in our body and they will educate the T cells and B cells to mount a response. So this is the vaccination approach. Another thing is like since as I was saying, cancer develops inside our body. And one of the mechanism by which our body is not able to fight cancer is because since it is our part, so our immune system is programmed to ignore our own body host cells. So essentially, like if our immune system starts attacking our own cells, then we will not be existing. So there are approaches, means so essentially like we don't have a good anti-tumor T cells in our body to fight cancer. But you can now create these cells. You can engineer these cells by different approaches. So you essentially take these lymphocytes, and there are different approaches. I will not go into detail, but let's discuss on this PCR and CAR engineered T cells. So essentially like these CD4 and CD8 T cells that I was talking about, they recognize the target through a specific receptor, which is commonly known as T cell receptor. So if you have a antigen-specific T cells for a cancer or antigen-specific T cells for a COVID, you can isolate the functional T cell receptor out of those cells. Now you can clone it in a vector, plasmid or a viral vector, which is harmful, means made harmless, and then it will be expressing your T cell receptor for that particular target. And then you take out the cells of this individual's body, and then you can engineer them to express that particular receptor, grow them in large number, and then put it back into the patient. So this is the mechanism by which this is approached. So this is the summary that you take antigen presenting cells and then culture them in the presence of the peptide, and then grow these antigen specific T cells, take out a receptor and then express it on a CD8 T cells, so they will become like effector. You can also put it on a MSC class 1 receptor on a CD4 T cells. So this is some of the work that we have done and then published quite a few papers. But whether it's this approach or another that is a chimeric antigen receptor, whereby you make use of the antibody and then couple it with the TCR. And so you make a construct, and then you take out a normal cell which are not antigen specific or that disease specific. But then by just expressing this molecule, you make them that disease specific army, grow them in large number, and then you can put it back. So I will just briefly take you through some of the data points so that you understand the science behind it. So these are some of the real cells which are in a cancer patient, and these are antigen specific T cells. And we generated a single clone out of that. And then by just showing them the target, they express certain biomolecules. And by that, you can check whether they are specific or not. So after we confirmed it, then we isolated this receptor by uh, technology, which is known as race. Different, everybody knows about the PCR. So it is essentially a modified form of a PCR, whereby you make specific primers and you can isolate them. So we identified two receptors, alpha and beta chains. So TCR are comprised of two chains, alpha and beta. They come together, and that's how a functional TCR is built. So we isolated two TCRs, and then we cloned them in a lentiviral vector, and then we express in cells. So we could see like we could grow a functional TCR in these cells. And then we wanted to test, okay, you have now built a off-the-shelf reagent. So now this is sitting in your lab, and then you can take anybody's cells, and then you can see whether you can create anti-tumor T cells or not. So we took normal cells and then expressed this receptor, almost 12% per antigen specific. And then we wanted to see whether they have become specific or not. So you show them the antigen. So we took a cell line, T2. This white one is not expressing any antigen. So this is a white. And then M1 is a right antigen. M3 is a wrong antigen. So these are small peptides that we put in. So once you put a right antigen, then only the response was seen. And this was almost like a helper kind of a response. So this was against cell line, but this is against the tumor cells. So this could detect the tumor cells and effectively express these molecules. So this was just showing that you can engineer a immune response which is tumor reactive. And then not only they produce cytokines, but they could also kill the target. So this is we are showing lysis of these tumor cells and also T2 cells. So once you show the right antigen, then only the killing is happening in a dose dependent manner. And this is against the tumor cells, since these cells naturally express some antigen, so they also get killed. But once you put the peptide, the killing is increased. So that's also a dose dependent. And as a positive control, we used natural CD8 positive T cells in there. So the summary is like you can engineer cells to fight off a disease and which they were unable to fight off before. Then we dissected the mechanism also, whether this is the traditional death mechanism or not. So there are some effector molecules known as granzyme and perforin. So we confirmed that the killing is through that traditional route. How we confirmed it? So these molecules reside in certain granules in the body. So once these cells detect their target, these granules gets released, and then these granules enter the target cells, and then they can kill those target cells by activating certain proteases known as caspase. So this is a red line is release of these granules. So we confirm that, and now of course as a positive control we use the traditional CD8 T cell. And if you remove this release of the granules by treating these cells by EGTA or by blocking the interaction of those MSC molecules, the killing was blocked. So that was our confirmation that the killing was happening through a traditional route. So this is the summary of the first part, which is essentially like how to engineer a good immune response. And the similar approaches are going on in different routes for COVID also. So you can essentially take CD4 T cells, and we converted these CD4 into helper CD4 cells, and they can boost the magnitude of the response because they were producing these helper cytokine, and they were also able to directly kill the tumor cells. These are some of the publications that we have published over the year. And our collaborator did the in vivo tumor targeting of these cells. So essentially engineered T cells were introduced into the animals in which the tumor was implanted. And then these cells were able to cure these animals effectively. And then these cells could go to the tumor site. So that is the in vivo efficacy. And there are A lot of clinical trials at this point going on in the Western countries, and some of them have also received approval, CAR-T, for example, for treatment of different cancer. Now coming to the stem cells, and what are these cells, and why you should know, and what are the different approaches that are being developed in a sense. So it means stem cells based regenerative medicine aims to utilize adult as well as pluripotent stem cells of embryonic origin, continuation of content of the youth, for replacing damaged cells, tissues, and organ systems of the human body. So essentially, like stem cells are nothing, but it is the story of you, story of me, part of your mother, part of your father comes together, and there comes a complex body, which is we sitting in our own respective yards. So this one single cell, which is a fertilized egg, forms a complex body which has lots of different organ systems, lung, liver, brain, heart, kidney, and they all have the same nucleotide sequence, but they have different morphology, they have different function, and they all function perfectly in unison without interfering onto each other's turf. And this is the beauty of nature, how this whole system is created from one single cell, and this gets developed into different functional cells. How does it happen? Like during the course of development, there are three germ layers essentially that comes into picture: ectoderm, mesoderm, and endoderm. And these ectoderm, mesoderm, and endoderm, they form different organ systems. Skeletal system comes from ectoderm, skin comes from ectoderm, brain, and then muscles, and then kidneys, heart, they come from mesoderm, and our gastric signal system comes from endoderm and all this. And during the course of development, like one single embryo, when it is developing, then for example, like your blood is developing, or your heart is developing, your kidney is developing. So that comes, there comes this tissue specific stem cells. So this is what I was talking about, like when I was saying like how these different cells sustain in our body. So you might have seen somebody losing nail, and that nail is, doesn't mean that that person will not grow the nail again. Your nail grows back up as long as you don't cut the toe. So because there are some nail specific stem cells sitting there, which will then grow a new nail. So there are stem cells in our body which are known as adult stem cells, which are associated with different organ systems. Your skin get scratched, your new skin comes back up. We time to time have to have a haircut because there are hair specific stem cells lying in our those part of the body. So there are some reservoirs of stem cells through which these different replenishment of old and dying cells take place, and the development of these different differentiated cells from early embryonic stage is known as differentiation process. That's how like our body develops. And these organ specific stem cells are known as hematopoietic stem cells, which are essentially responsible for blood, and they reside in the bone marrow. Mesenchymal stem cells, they form different parts of the body, and different types of cells. They can reside in the fat and also in bone marrow. Neuronal stem cells associated with neuronal tissues, epithelial and skin stem cells also. So these specific areas where our stem cells reside, they are known as niche. And they are essentially like some reservoirs where they can maintain their number also. And when there is a need, they will differentiate into different types of the blood cells. So if you go for sometime a blood test, if you have gotten a disease, then you get a blood test, and then it gives you a number. So many macrophages, so many neutrophils, so many T cells, so many lymphocytes. So this is all these blood specific cells are coming from one progenitor, which is known as hematopoietic stem cells. So this is what I wanted to emphasize, like there is hematopoietic stem cells, and these hematopoietic stem cells will have to maintain its own number also, and this will have to form daughter cells. And daughter cells can be neutrophil, daughter cell can be macrophage, daughter cell can be lymphocytes. So this whole process is going on in our body all the time without you knowing it even, or noticing even. And this is the job of adult stem cells. And I will not bore you with the intricate details, but this field is progressing quite a bit. And now starting from early 1962, when the first somatic cell nuclear transfer approach was developed, to 2007, when these donor-specific induced pluripotent stem cell lines were generated, which brought in 2012 Nobel Prize for Shinya Yamanaka. So this is the technology, different technologies by which you can convert a normal cells into the donor-specific stem cells. Why I'm telling you is because like sometime like a individual may have a bad liver, sometime an individual may have a, bad heart. So these stem cell based therapeutics can be of great use to help those individuals. So there is a big scope for developing these individual specific stem cell lines. So this somatic cell nuclear transfer, so you can take a somatic cell, remove the nucleus, and then transfer this nucleus into a nucleated egg and then generate this. Or you can take early stage embryo, and then from there you can take out pluripotent stem cells. But this is the technology that I was talking about. You can just take a bunch of genes, 4 or 5 genes, and put it in a normal cells. Essentially, I can take your cells, express these genes, and then make a iPSC, which is specific to your own body. And it will be sitting in the lab. Whenever you need a particular type of cell, we can generate that. This is the example. So we took one individual's blood cells, expressed certain molecules, and this is a iPSC clone that was generated for that individual. And this is how stem cells look like. So you can essentially generate individual specific stem cells. These are some of the technologies by which you can characterize. So by looking at different gene expression by RT-PCR, or there is a technology known as FACS, fluorescence activated cell sorter, by which you can see the surface expression of molecules, or you can purify them. By microscopy also you can generate. So these are some of the technologies by which we confirmed their pluripotency nature. How do you use these stem cells for therapeutic applications? So essentially, like you take patient's cells and then express these factors, and then so that you can generate these stem cell clones. And if that individual needs nerve cells, then you can generate nerve cells. If that individual needs blood cells, you can generate hematopoietic stem cells. Or disease-specific individual you are taking, Alzheimer patient, you can generate Alzheimer specific neurons, and then you can test the drug screening drug library to identify new biomolecules. And if you generate these functional cells, you can put them back into the patient, and that will treat the disease like so. So I'll just briefly touch upon like what we have done in this scenario. We generated those iPSC clones, and then from there we generated these functional antigen presenting cells. What I was telling you in the beginning was antigen presenting cells are something that you can generate vaccines from. So essentially, like we have generated iPSCs, individual specific, and then by these different steps, we first we program towards monocytic lineage, myeloid lineage, and monocytic lineage, and then dendritic cells. And these are actual these dendritic cells that they are looking, and their key property is once they see the antigen, they eat it up. So we put a dextran beads which are labeled to different dyes. So you can see like these cells. So this is the control. None of the cells are towards the right side, but once you see these test cells, they have engulfed these beads. So essentially, they are phagocytizing the antigen, and we confirmed it under the microscope also. So they are essentially functioning as true antigen presenting cells. They can acquire the antigen, and we have published this work. So you can, anybody interested, they can go through it. So they can present the antigen to T cells also. So this is about the how you can generate individual specific stem cells, and how you can generate individual specific cells for different therapeutic options and all those things. Now, to come to the materials and material science, because this is a forum for materials. So as I was talking to dendritic cells and T cells, so dendritic cells acquire different antigens through different route. Why I got interested into this material sciences, because like when we have to introduce different genes, then you have to use different lipid molecules, liposomes and all those things, and also nanoparticles. by which you can deliver these different effector molecules for the delivery of the genes and other effects. Like so, in essence. So that's how we got interested, and then we collaborated with Dr. Watts, and then a project took off. That was how we can generate a good skin cream that will fight off infection, and that will also be able to rejuvenate your skin. So we have created a skin cream, and this work, as I was talking about, was done by Shruti, a MSc student, and means so this is the cream. Like we are further defining it, but like this is our first prototype that we have developed. On the left side is a normal, and on the right side is a nanoparticle infused cream. This is the patent application that we have filed in means Indian patent. So this was done by green synthesis manner, whereby we use cucumber extract for reducing these means chemical reaction. One of the key feature was this our skin was antimicrobial, anti-inflammatory. So essentially, like we are seeing these days, as a COVID arise, then we thought of repurposing our cream also, and we are working on it. So because like every time you have to take sanitizer and all those things, but instead of that, like if you apply a cream which can be antimicrobial, anti-inflammatory, and keep your skin also rejuvenated, and I think it will provide you a better protection in case you don't have access to this sanitizer all the time also. So that's the purpose behind it. So Shruti started this synthesizing these nanoparticles using this cucumber extract. So these are some of the reaction steps, and then these are nanoparticles that she prepared. And this is the XRD, like for their physical characterization. Zeta potential also, like so essentially these are some of the techniques by which you can see their stability, see their charge, and you can also see, like if you are preparing nanoparticles, what is the whether they are essentially in a nano range, 1 to 100 nanometer size range or not. So these are some of the techniques that are used. So this is a SAM that was done on this nanoparticles. So we confirmed their biophysical properties and all those things. Then came our means basic nitty-gritties. That is whether these nanoparticles have antimicrobial properties or not. So this is the sample, which was essentially showing quite a bit of antimicrobial property. And of course, you have to use a positive control, means antibiotic or something like that. So, but essentially, like they were exhibiting antimicrobial property. And then the next thing is, if you want to use it for human application, whether it is toxic or not. So we confirmed that our sample was not non-toxic. So we took human blood-derived cells, and then co-cultured these blood-derived cells with these nanoparticles, and look for hemolysis. We did not find much of a hemolytic reaction in there. So we determined that these are safe in the nature. And that's when we moved to preparation of this nano particle infused cream. And once you create this screen cream, then you have to see whether your cream is stable or not. So we ran through different centrifugation forces, and this was able to hold it itself. And also this withstood the temperature also up to like 40 degrees centigrade very easily. So that showed us that our cream is stable enough. Then in our cream was also infused with the antioxidant properties. Cucumber itself has some of these properties. They get transferred once you are using this green synthesis manner, but we also infuse these as a vitamin E within this cream. So this cream had antioxidant activity, because we wanted to have this, because free radicals are one of the key challenge in our body, and these free radicals are also essential for means also precursor for mutagenic activity. In the beginning, when I was talking about cancer development, so mutations accumulated as a result of higher inflammation and higher this anti in this free radical activity can result in accumulation of these mutations. So this cream has antioxidant activity, which is good, and then this has good enough moisture content, and this is the ash value in there, and then beta carotene is a skin rejuvenating agent, so we could have good value of beta carotene also, and it was stable over the duration that we tested. And what was more important is that it had a good SPF value of 37. What is a SPF value? This is known as a sun protective factor, sun protection factor. In India, especially in the northern region, we don't go much for this skin bathing or sunbathing. But like a lot of people in the villages, they go for sunbathing. And in the Western countries, especially like they go for these beaches, and they have much lighter skin tone. And skin cancer is a big challenge. So to protect the skin from getting these exposure, means lot of exposure to UV rays coming out of the sun, and they apply these skin creams, which have these standard SPF value, which should be about 30. So our skin cream has a value of 37 SPF. So this can be also used as that sun protective skin cream. So essentially, like we have created a skin cream which you can repurpose it for antimicrobial properties, for skin rejuvenation, and also for skin protective ability. And now, like since I come from a stem cell means area also, so we are trying to develop another variant by which we can infuse them, because in our under our top layering of the skin, there are skin specific stem cells. As we are aging, some of you might have seen your grandfather, grandmother, they developing wrinkles. So if you can activate the stem cells which are lying below the skin as you age, then your skin can be kept healthy, and then this wrinkle development can be reversed or reduced. The kinetics of it can be reduced. So that will be our next phase of the development of this work in this. But like as I was saying in the beginning, intellectual property needs to be protected. So we have protected our preparation by patent also, and after that we have published also. So if some of you may think or come to know that once you file a patent, you cannot publish. So this is the example that you publish it, but not before filing the patent. Once you file the patent, secure the priority date, then you can go ahead and publish your findings also. Now, the another work that I want to briefly touch upon is in this COVID scenario is development of a affordable, reusable, biodegradable, antimicrobial, multipurpose face mask. So you have different face masks in the market. When we started this project, they were very costly, and they were of one application only. But now there are much more variants available. So as we know, like COVID develops as from a coronavirus, and the mode of transmission is through respiratory route. So if you come in contact with somebody who is infected with the virus, they are breathing, and with their breath, the virus gets released. And if you are close enough that you will just take the air in your respiration, and the virus enters essentially your body. So that's the purpose of introducing these face masks. That if you have a face mask, the droplets that are coming up in through that individual, they will get a physical barrier. And on top of it, apart from a normal clothing mask, if you have a layering which has a antimicrobial properties, that will essentially kill those viral particles. And also there are different other pathogens which are present in the this air. So this way you can, this is the purpose of the face mask. So my slide got frozen. Hopefully it will move further, and then we will go further. So this was the basic idea, like that we started by taking this this project. Let me wait for the slide. This is, I think my computer is taking a little pause. So which I think gives you also a pause for a minute. We have touched upon that many aspects. So this is our first crude prototype that we developed, but this is the idea. So you make a two-layer face mask, and then which will have a replaceable insert, and that insert could be your nanoparticle coated layer, which will fight off the infection, or it can be N95 grade insert, and or it can be infused with certain essential oils or something, and it can provide you some aromatherapy benefits. And moreover, like now we are also thinking about making a disease specific mask, because asthma is one of the challenge, and asthma inhaler is a big cost for individuals in not only in the US but in India also. So you can infuse different drugs within these inserts, and then this can be a disease specific face mask. So the reason why I'm showing you the secret right now is because we have also filed a patent for this. So we are not showing you prematurely. So if you think that you can steal the idea, my apologies. I'll come after you filing a lawsuit. So and don't get bothered about it. But I'm just saying, like whenever you do some science, protect your idea. So we have filed a patent, and our team includes from AUH, as well as Dr. Shatindra Sharma from JNU. And of course, Monica is there. And we had another patent which is associated with this project itself. So this was our first patent in this line, whereby our initial idea was we wanted to create a fabric which will withstand stain. So essentially, like which will be able to clean itself. So since nanoparticles exhibit antimicrobial properties, and some of these nanoparticles can degrade dyes also, as we have seen, and they can, I mean, some nanoparticles can quench the dyes but in a gel form, as we were seeing in the beginning, but like some nanoparticles can degrade the dyes. So means the basic idea was, since nanoparticles can degrade the dyes, so if a stain is coming to the cloth, then this will not gets permanently anchored to it. So you go for like outdoor activities, tents and all those things. So those clothings can be infused with these nanoparticles. So we have two patents, and then we are working on developing those prototypes and all those. But restricting to the face mask itself, so there are different face masks available, N95 grade, or some have a biodegradable, means antimicrobial properties also. But if you see all these face masks, like surgical face masks or N95 grade face masks, what do you do after that? Most of them are non-reusable. You just throw them in the dustbin. And if some of you might have noticed on the road, there are lots of animals loitering around. And as we know with the COVID came from the bats. And now we know like this can go from human to animals also. And animals can serve as a reservoirs. So the in a population of 1.3 billion people, if you are throwing a billion face masks over a duration of a year, or even less than that, some of it is going to go into the animals, and that means that the virus is going to sustain in the environment, and then this disease is never going to go away. Never going to go away, because this virus is prone to mutations. We are already hearing there is a different mutagenic form of this virus that has developed, and if it sustains in an animal host, then the mutation cycle will create even more dangerous variants, and that will become a problem. So the solution is you use a reusable mask, and you wash it off with the detergent. The virus is clipped up, and then you can reuse it. And once you throw it away also, just throw it in a proper manner, so that the virus cycle is broken in a sense. So this is the purpose of this antimicrobial face mask that we envision, that this has to be a reusable face mask. So we had this is an example how the nanoparticle coating was done. The cloth was treated with the nanoparticle, and means our cross-linking was done in a such a way that this also provided the hydrophobicity properties to this nano-coated cloth also. So essentially, like if you are going through a rain, then still this your antimicrobial layer will be able to protect you against any droplets that are coming with the viral particle. And as I showed you with the earlier nanoparticles for the skin cream, we again did the biophysical characterization of all these nanoparticles by different techniques, XRD, and then UV visible spectra, Raman spectra, and then of course TEM, so to size them up. So all these were confirmed, and then their photokinetic activity was determined, their ability to break the dyes and all those things. And once you have coated this means cloth with the nanoparticles, we wanted to see like whether you wash these cloth, whether these nanoparticles come off also or not. So which is very important, like how long you can keep it coated to the cloth, because it is very important, because if you are coating the nanoparticles on a cloth and you are putting it next to your nose, if you are infusing those nanoparticles, it is going to be some harmful, unless you use inert nanoparticles, of course, like silver or gold, which we are working on. But you don't want accumulation of nanoparticles in your body. So we confirmed that after once you wash it, there was no leaching of these nanoparticles from the cloth. And of course, we confirmed the antimicrobial activity of these nanoparticles. So these were exhibiting good nanoparticle mediated antimicrobial activity, and we also tested this toxicity by again hemolysis. So we took the blood and then exposed this blood to the nanoparticles, and no killing of the cells took place. So they were essentially safe. So among the potential application, they can be used for infectious diseases protection. And once COVID is also gone, then also like there is a underlying issue of air pollution. So the face mask will sustain for means protecting us against air pollution also, and there are lots of other pathogens. So that will be very much useful in that scenario also. And face mask for anti-inflammatory drug delivery, aromatherapy. So these are some of the applications. In the last slide, I would like to conclude by acknowledging my collaborators over the year. So as we, Dr. Mangla in the beginning said, like I was at University of Connecticut Health Center for almost like 18 years. And they're my collaborators for Dr. B.J. Mukherjee, Upendra Hegde, Shikhar, Nitya, and Swagatam was a postdoc fellow, and Deepika Raja also worked with me. And we had collaborators from UCLA, Dr. Jamie Kahneman, Anthony Ribas, and from Caltech, we had Dr. David Baltimore, who actually was the one who discovered this reverse transcriptase enzyme. And I was born in 1972, and Dr. David Baltimore got Nobel Prize in 1972. So you can imagine shelf life of a scientist and the productivity that a person can sustain over the years. So he's still very active, and different means parts of science, and going great guns still. And at AUH, we have collaboration with Dr. Satish Sardana, who is the director of pharmacy department. Then Dr. Monica Vass from ASAS. She is an excellent material scientist in her own rights. You will hear her some other day. And Dr. Shruti and was working earlier on this skin cream project, and Neha is working now on this face mask project. And then we have a collaborator from GNU, Dr. Shatendra Sharma. So with this, I would like to thank again all the organizers, especially Dr. Mangla, for making me part of this meeting. And I invite all the questions that you might have. And with that, I will stop.
Thank you, Dr. Arvind. Thank you. May I have the queries from audience? Any question or queries if they want to have? They can unmute themselves, and they can also raise their hand. We have one hand raised from Ritika. Ritika, you can unmute yourself.
Good afternoon, sir. Good afternoon, ma'am. Sir, as you said that antimicrobial face mask is useful, but how long it we can use it? Like, is it having any temperature range? Like if we expose it, if it expose in a high temperature, like in summers, will it cause any harm to our health?
Yeah, so basically, like since we are coating this insert with a nanoparticle, and then they are quite stable at a higher temperature, even in India also, and at those temperature also, like 40, 48, and even then it doesn't lose its antimicrobial properties, so they can be used very safely in a sense. So, and as I said, like these nanoparticles don't get bleached out of the fabric, so they are safe to wear also. But like some people may have concerns about nanoparticles, so we are going for natural preparations of antimicrobial nature also. So we have different inserts. So essentially, like the basic idea is, if you are interested in going out with a nanoparticle insert, so you go out with a nanoparticle insert or N95 grade insert, you come back home, and then you feel like, I want to have an aromatherapy insert now. So without replacing the different mask, you just replace the insert only. So this is that way you can just change the application. But short answer to your question is, this will sustain its antimicrobial property at higher temperature also.
And is it safe for every age group? Like, can we use it for like smaller children's also?
Yes, yes, yes. So it is safe for because you will not be infusing it, and we are making sure, like further to reduce inhalation of any nanoparticle from there. So we are putting a membrane in front of these means nanoparticle coated fabric. And as I said in the means earlier, that we are taking essentially plant derived certain essential oils, which also exhibit these antimicrobial properties, and also inflammation removing properties. So that will make it means even more safer for all the different age groups. So the prototype that we are developing, I showed you the adult version, but like in the next time when you see our final product, I will give you like a child version also of that. Okay.
Sir, and as you said, the cream, the substituted,
Yes.
the cream you said,
Yes.
and so how it works? Like, is it safe against viral infections also? Like you
Yes.
said, it's microbial properties, antimicrobial properties it's having. So
Yeah.
is it
Yeah.
safe against it also? Viral.
So basically, like the virus gains entry through your the extremities, like respiratory routes and all those things. And another thing is, like it can sustain to certain surfaces also. So and there are these like a clove and other essential oils that we have. They have antiviral properties also. So we are infusing not only these nanoparticles, but some other factors in there which have antiviral properties also. So that's how we are making is our initial version one to suit it to COVID scenario also, to it off with the virus. Okay.
So it will depend on the long-lasting nature on the skin. Means if
No,
it will
means
not.
so essentially, like when you go out, you use sanitizer. There is a alcohol in there, and say for example, like you are traveling, you don't have any access to sanitizer. So in that case, you can apply the skin cream, and skin cream can provide you protection for a longer. So whole day you can get protected by providing you a means this antimicrobial and also antiviral property, and it will keep your skin also healthy against your UV rays infused danger or something like that. So it is like essentially a normal skin cream, which we have repurposed it for the COVID scenario. So you some people use moisturizers and all those things, and they are quite safe to wear, right? So they don't damage your skin. So we are just infusing these antimicrobial and antiviral properties in this. And later on, our version 3 will be to activate the skin resident stem cells for further means applications.
Okay, sir. Thank you so much.
Sure. Any other
Thank
question?
you, Ritika. Any other question from the audience? Sir, I have one query.
Yes.
Like you explained the use of copper oxide nanoparticles and some other metal oxide nanoparticles.
Yes.
And we are somewhere we are also concerned with the toxic nature of these metal oxides.
Right.
So is it safe to use these kind of nanoparticles in cosmetics? Because although we are applying them on the skin, but somehow in some cases it get also engulfed, maybe because of any reason. So is it
Yeah.
safe to use that?
So that's why, like I said, like means copper is being used for a time immortal, like for utensils and all those things. So copper is one of the nanoparticle that has all means, or one of the surfaces on which virus survive the least amount of time also, and it has antimicrobial properties also. So the copper is the safest, and also like silver and gold, they are already being used for different biomedical applications. So copper and gold nanoparticles, copper and gold labeled antibodies, they are already being used for biomedical applications like so. But like I fully point that if you are talking about respiration route, so that's why like we wanted to ensure like whenever we are cross linking these nanoparticles, they shouldn't leach out. So that is the safety concern that we are trying to ensure. And then on top of it, like if we can reduce the further accumulation by putting at additional filters in front of before your nose and this nanoparticle infused fabric, so that will further reduce it. So I'm fully aware of that concern, and like concerned about that particular issue. And then we are trying to make it safe, and that's why like the reason in the version 1 that we are going in, we are essentially going in with this plant derived products which will have no nanoparticles also. Some people who are averse to nanoparticle use, so you can opt for without any nanoparticles insert also. I'm fully aware, like a lot of people have already gone into the market, like which are claiming that they have nanoparticle infused nanoparticles or infused masks and all those things. But I am concerned about the safety profile as they have done or something like that. So that's why we are taking all the precautions to roll out the product in the best possible manner.
Thank you so much, Dr. Arvind.
Sure.
Anybody with a query? They can raise their hand or they can unmute themselves. So I think Dr. Arvind, you explained very well about these cosmetics we can develop. And I remember the time when this Aatmanirbhar Abhiyan just started. The key point of this Aatmanirbhar was to be Swadeshi, to use Swadeshi. And FMCG sector is one of the biggest sector where we are importing, or you can say where we are depending on the foreign companies. So if our scientists are developing such kind of products with good quality and these features, I think we all will be Swadeshi very soon. And this Swadeshi movement will be a big backbone for this Aatmanirbhar Abhiyan. And you explained your research, which is developing like antimicrobial face mask and others. So I think this is one very uh, strong foundation for developing a nation with the Swadeshi products. And
Right.
we will be able to see more towards it rather than depending on the companies which are foreign based. And sir, you explained the things in a very simplest way so that each and everyone can imbibe these things in a way like they can practice it in their own laboratories. And I hope you don't mind if anybody approaches you for doing something more in this field. And
Sure.
and I hope that this collaboration with you of our department will go in a long way. So
Sure.
with these words, sir, I am indebted for with you. Like you have spared your valuable time and delivered such a nice talk to our participants. Thank you very much, sir.
Thank you very much, Dr. Mangla. It will be my great pleasure to build more collaborations, and students are more than welcome to write to me, to come and meet me, whichever way they feel comfortable. If they have some ideas, because the reason why I was putting up more emphasis on patents, because after I came over, I'm also a facilitator of patent cell here. So if you have any idea, you want to discuss any idea of whether it's a patentable or not, so I'll be more than happy to give my advice. So by all means, I'll be more than happy to contribute in whichever way to the growth of the department or growth of the students.
It will be my pleasure, sir. Thank you so much.
Thank you.
So I thank both the speakers of this day. And with this, we are coming to the end of this first session of this day 5. And we will meet again at 2 o'clock in the next session of this day 5. And by tomorrow, we will be having the last day, day 6. By morning, we will be having the sessions. And by after post lunch session, we will be having a valediction and a surprise session on this Aatmanirbhar Bharat. So thank you so much. I request all the participants to join back again at 2 o'clock. Thank you. Thank you so much.
Sure. Thank you very much. Bye-bye.